Okay, everyone, we're going to start this session, Genomic and Precision Medicine for Therapeutic Research and Development. We have three outstanding speakers today, and the first one that I'd like to introduce is Dr. Lyndon Mitnaul. Dr. Mitnaul is the Executive Director in the Regeneron Genetics Center at Regeneron Pharmaceuticals in Tarrytown, New York, where he establishes and manages multi-organization, large-scale, diverse collaborations and education and training initiatives. In 2023, Lyndon championed the formation of a new initiative, Together for Change, to increase the number of underrepresented students in STEM and to significantly increase diversity in genomic databases being studied. Lyndon?
Thank you for that introduction, and thanks to the organizers of this wonderful meeting at this beautiful country. My first time in Singapore, but definitely won't be my last. I have the pleasure this morning, today, to tell you about a new initiative called Together for Change. And I think this initiative is a great example of doing good science and doing science for good. And these are principles that we have at the Regeneron Genetics Center as well. So let me tell you just briefly about the Regeneron Genetics Center, and then we'll get into Together for Change. The RGC applies large-scale, fully integrated human genetics to advance science and guide our drug development programs. We use human genetics to identify new indications for drug programs, to identify new drug targets and pathways, and link our human genetics to our mouse genetics technologies like Velocigene. And we use human genetics to utilize pharmacogenomics for patient stratification to implement genomic medicine into health systems. We don't do this in isolation, but we do this in collaboration. We have over 130 collaborations around the world in over 23 countries, and those countries in blue are where we have or had active collaborations, and those in green are those where we aspire to have collaborations, working towards collaborations, including here in Singapore. So I would love to speak with you if you would like to collaborate with the RGC. With our collaborators, we've made a lot of contributions. We've sequenced over 2.5 million exomes, individual exomes, and over 500,000 of these exomes come from people of underrepresented populations. We've identified 20 novel genetic targets that we're working through the pipeline now and have contributed to over 30 programs in our clinical programs. And as I mentioned, we help to advance science, again, with our collaborators in publications, and these are just a few of the publications listed here that we've had more recently. So, when you think about cohorts to the clinic, we have one example that I would just like to tell you about today that started back in 2021, where we sequenced over 640,000 people to identify a very rare variant in a gene, GPR75, that protected against obesity. Obesity is a big problem, as we all know. And people having one copy of the active GPR75 gene has a lower BMI and also weigh about 12 pounds less and have a 54% lower risk of obesity versus those that carry two active genes. This protective variant, and by the way, that's data there, this protective variant is very rare. It's in about 1 in 3,000 individuals that we've sequenced. And, I know it's hard to see on this slide, but the last two cohorts, large cohorts that we've used in this analysis, the UK Biobank and Geisinger Health Systems, they had a signal, but it wasn't significant. And we kind of heard this today about biobanks and the need for more biobanks and large-scale sequencing projects. So when we, in more diverse large-scale sequencing projects, when we added the Mexico City prospective study to this analysis, then we saw significance of this signal. So just wanted to highlight more diversity in our biobanks and more large-scale biobanks around the world. We took this genetic finding into our mouse using velocigene technology, knocked out the mouse, and we can see that when we put them on a high-fat diet, the mice had a gene dosage effect of less weight gain. So now we're hoping to take this cohort to the clinic through using different therapeutic modalities. You know, a classic example, though, is PCSK9. This is University of Texas Southwestern group that did a population study of African Americans and non-African Americans in Dallas, Texas, and the Dallas Heart Study, where they recruited participants, sequenced them, and looked at a lot of cardiometabolic traits. And they saw that African Americans, there were about 3,300 African Americans in this analysis. When they looked at the cholesterol levels and the LDL, bad cholesterol levels in these participants, they saw less LDL cholesterol in carriers of a very rare variant in PCSK9 at this time. And you can see from the plots here on the bottom that those that carry the variant had less LDL cholesterol, but also they were protected from cardiovascular disease. This led to a lot of researchers to study this pathway, and they realized that PCSK9, this protein secreted by the liver, would help to degrade the LDL receptor upon endocytosis. So we and a whole lot of other companies have worked on this pathway and worked on, making antibodies and silencing RNAs to, reduce the amount of PCSK9 binding to the receptor. And I'm just showing data from our phase 3 trial here on the top right, where you see a 50 to 60% reduction in LDL cholesterol when you infuse this antibody, aluracimab, into patients. So again, going from cohort to clinic, and I think this is a classic example of what we all would like to do here. In addition, this study and this cohort has really, been very beneficial to a lot of researchers in that, over 230 articles were published on this cohort, and this led to a lot of grant funding to researchers studying this pathway. So how can we continue to do good science like this and do science for good? I think there was a call to action in 2021 in this publication that talked about concrete steps to diversity, to building more diversity in our STEM workforce. From this graph, you can see that Black Americans, Latinx, and indigenous populations are underrepresented in the STEM workforce. That's the bar graphs here. But more strikingly, their progression declines as they go from graduate school, from undergraduate to graduate to faculty. They actually decline. So that's a big problem. Secondly, we just wanted to look in the literature and see sort of what publications have been, published on genetics in a 10-year span from 2011 to 2022 shown here. There's about 30,000 publications that were published on genetic type studies. And about 4% of those genetic studies included people of African ancestry. But when you break down the data a little more and look at the schools, like our historically Black medical schools in the US, and say how many papers were published from those schools, about 80 papers were published, or 0.2% of the publications. But when you look at those publications further, 50% of those publications included people of African ancestry. So this shows that researchers have a personal connection to their ancestry and country of origin. So what concrete steps can we take to diversify both the educational pathways and support students and mentor them through their pathways so it doesn't decline? And secondly, how can we build more diversity into the schools that are studying genetics and bring more genetics research to schools that are underrepresented in this field? We think it's through this initiative, Together for Change. This is a 10-year commitment from a lot of organizations to support communities, science, and society. And there's a lot of organizations that are already a part of this, and I'm hopeful that others will be joining us. The Together for Change initiative helped to establish a new 501c3 nonprofit organization called the Diaspora Human Genomics Institute. This is, again, a nonprofit organization that will be managing the Together for Change initiative, and it was chartered by our foundational HBCU partner, Meharry Medical College out of Nashville, Tennessee. I'm proud to say that the next two speakers are also a part of this initiative, them and their organizations, AstraZeneca, Roche Genentech, and also Novo Nordisk. So thank you guys for being a part of this. So what are we trying to change? We're trying to change healthcare for everyone, just like in the PCSK9 story. And we're going to put a focus on people of African ancestry. And it's going to be through an international genomics and equity initiative. So there is a genetics component to this, but there's a lot of education, training, mentorship built into this also. So we're trying to address two systemic things that need addressing in the Black community. And that is building more diversity in our STEM careers. You know, only 9% of our professional STEM careers are held by Black professionals. And this is closely tied to the educational system where these students and people come from. Secondly, we want to diversify genetics. We've heard this many times in this meeting that, you know, we need more diversity, and especially in African ancestry genetics, and where only 2% of the genetics we study today come from people of African ancestry. Now, I know on the previous slide and on this slide, there's a lot of other colors that need improvement here. Don't get me wrong. I'm all for inclusivity and equality here. But we have to start somewhere. And I'm hoping that this Together for Change initiative is a catalytic model or blueprint for other, you know, for us to do this for all of these other groups here that need representation and help. So how are we going to do this? We have formed a unique public-private partnership and collaboration ecosystem that, again, is being managed by the Diaspora Human Genomics Institute. We have our foundational HBCU partner, Meharry Medical College out of Nashville, Tennessee, but we're also inviting all 107 other historically Black colleges and universities in the U.S. to join. We're also asking organizations in Africa to be a part of this, so we've already reached out to a lot of different organizations there, and we need our majority institutions to be a part of this too. We need biopharma to be a part of this. If we're going to take cohorts to clinics, you know, biopharma has to be a part of the equation here. So I'm, again, very grateful of the companies that have joined, and we're constantly recruiting other companies to join us in this effort. And, you know, together, this ecosystem that we've created, we're going to be doing a lot of community engagement, collaborative projects, and these collaborative projects will lead to multi-institutional funding for schools that typically don't get funding from genetics research. By us all coming together for 10 years, we're going to be having a lot of mentorship and training opportunities, knowledge sharing, professional development, and career opportunities for students as they see different pathways that they can go into, academia, industry, and others. So we have 5 main goals that we want to address in this initiative. One, we want to empower communities on health and medical research studies. We heard about this yesterday and earlier today, that, you know, we got to build more relationships and partnerships with the community to build that trust. Community listening sessions are going to be had so we can hear from the community of what their concerns are with genetics research and genetic testing. We're going to have community health events as well as our first health summit in Nashville at Meharry later this year. We want to deepen our community STEM education through specific programs that we've set up. For example, we're building a DNA Learning Center at Meharry Medical College where young students can come on site, do experiments in a lab-type environment, and be inspired to go into STEM careers. We're going to expand the genetic counseling workforce. Less than 2% of the genetic counselors in the U.S. are Black. So we think having the first joint HBCU genetic counseling program at an HBCU will improve this. we're also creating the first medical genetics fellowship program at a historically Black medical school. And, you know, we want to increase the number of African researchers that are studying genetics. And we're going to do this by sharing the data that we're going to create from this initiative exclusively with them and give them the advocacy and the power to share the data with whom they want to share it with. So it's not exclusive to our membership. It's exclusively given to our membership to share with other collaborators that they want to collaborate, put the power in their hands. Secondly, we're going to, you know, bring them fish, but we got to teach them how to fish. And we're going to have this training programs, workshops, webinars, co-ops, internships, you name it. We're going to train and teach them how to analyze these large data sets because they've never done it before. And last, we're going to diversify the genetics and research and participation by building these community partnerships, and then sequence 500,000 people of African ancestry and link their genetic information to curated and harmonized electronic health record information and other phenotype data that will be collected on those volunteers. We launched this last October at Meharry. We had very good reception from the audience that was there. And, you know, there were a lot of questions about this initiative from the public. And it really showed that, you know, African Americans really want to understand genetics more and to understand how it will affect their health. We did a artificial intelligence machine learning activity experiment looking online to see what sort of conversations that were being had about genetics research and genetic testing. And there were 650,000 discussions being had online at this time, with 50,000 of those discussions coming from people who self-identify as Black Americans. This is a summary of that data from the Black Americans. And quickly, if you see on the left here in green, about 20% of Black Americans had a positive view of genomics research and genetic testing. And this was down 1.5-fold versus the overall average. If you look on the far right in red, about 30% of Black Americans had a negative view of genomics research and genetic testing, and this was up 1.7-fold versus the average. And a lot of that negativity came from confusion, misinformation, mistrust of their healthcare provider, cost, and how it will help them. They didn't understand how genetics would be helping them. I think a big opportunity is right here in the middle, where you see 50% of the Black Americans here went online to understand what genomics research and genetic testing are and how it will affect their health. So I think this initiative is very timely because we built in funding to have these conversations and community engagement events with folks to explain genetics and how it will help them. So we're hopeful that we'll move this, you know, 50% and some of the 30% negativity into the green and have more folks to understand how genetics affects their health. So how are we going to get to 500,000? Well, Meharry will be doing some recruitment from their hospital system there in Nashville. We've been in conversations with a lot of folks in Africa and putting a big focus on East and South Central Africa. But the majority of the samples are actually coming from the US, from different majority institution partners that we'll be having in population cities where there's a large population of African Americans. And I'm proud to say that the RGC is performing all of the sequencing in kind. We're going to do 500,000 whole exomes and genotype by sequencing, and at least 50,000 whole genomes for this study, and use that information to build better imputation tools so that we can impute on this population. And as I mentioned, all of this data will be linked to curated, harmonized, curated phenotype data like electronic health record information and other phenotype data captured from the participants. You know, since the majority of the samples are coming from the US, we wanted to look at our first 50,000 African Americans in our RGC database. So this is just a quick snapshot of the genetic ancestry of the average participant in the different cohorts listed here on the y-axis. So there's 45 different cohorts here, and going on the bottom is the different ancestral or regional groups. And you can see that, like most databases, our database is very heavily skewed to European ancestry. But there are some that have African ancestry. But when you look closer at this data, you can see that the majority of the African Americans that we've sequenced in our cohort so far come from Western African countries. So again, we would like to fill this gap by sequencing more folks from North, East, and South Central African countries in combination with our African American cohorts in the US. Even with this data, though, you do see more variance in people of African ancestry as compared to the other ancestral groups, both total variance, missense, and putative loss of function. And when you look at variants of unknown significance, again, this group has more VUSs. So we're hopeful that when we sequence 500,000 participants of African ancestry, we'll make more discoveries as well as help to validate some of these variants of unknown significance. So we've created this unique public-private partnership. We hope that we're going to be doing some good science and doing science for good. We know there's powers in numbers, so we need you. So I kindly ask you to see what you or your organization can do to help support this initiative. But secondly, to maybe use it as a blueprint to help those other groups that need more diversity built into the genetics databases. On behalf of a whole lot of other folks and organizations, I thank you.
Thank you, Lyndon. I want to remind the audience that you can ask questions during the talk using the Pigeonhole app. The QR code is right up there. The passcode for that is PIC24. The QR code's right over there. It's right over there. And it's always up in this corner here, too. So you can scan that at any time with your phone. Our next speaker that I'd like to introduce is Dr. Guillermo del Angel. Guillermo is currently the rare disease head at AstraZeneca's Center for Genomics Research, and in this capacity, he's been leading and developing different approaches to use genetics, computational biology, machine learning, and data science to improve rare disease target discovery, drug development, and patient disease characterization. Before his current role, he held several positions at Alexion, AstraZeneca Rare Disease, since joining in 2015, and included heading the bioinformatics and data science team, as well as heading the external search and evaluation function. Guillermo, the floor is yours.
Thank you for the introduction, and thanks everybody for being here. And I especially would like to thank the organizers for giving me the opportunity of for giving this talk. It's also my first time in Singapore. This is a great place, and hopefully will not be my last as well. So I want to briefly touch upon how we're thinking about using large-scale genomics for drug discovery at AstraZeneca. And I want to give kind of 4 little vignettes. Given our time constraints, I won't spend that much time in each of these, but this is just to kind of show you how we're thinking about, in general, using human genetics in the context of drug discovery. So as we obviously know, human genetics can transform drug discovery. I mean, this is a very well-known fact. We have seen over and over again that genomics offers a massive opportunity to transform our pipelines and our drugs pipelines, because we have to be cognizant of the fact that most of the things that we try in drug discovery will eventually fail. I mean, it's a reality in this industry that probably 90% of the things that we try will eventually fail. And a lot of the failures, people have studied them, and they come from lack of effectiveness. And we have also seen that drug targets that have some kind of genetic support are more likely to be approved. And you can measure this in many, many ways. There was a recent paper by Eric Minnikel trying to actually do a better job at quantifying this that I would encourage everybody to read. But essentially, it underscores the fact that human genetics really should be and can be leveraged to actually improve the way in which we discover drugs. So about 8 or 9 years ago, AstraZeneca launched the Genomics initiative, which aimed to sequence 2 million genomes by 2026, and we're well on our way to do that. And we eventually ended up creating this integrated set of capabilities, not just around the pure genomic data, but actually really on developing the tools and the organizational capabilities of actually using that data as much as we could. So we started work on multiomics, on functional genomics, on actually connecting those with our in vivo and in vitro model generation capability. And actually, we ended up also leveraging quite a bit of internal and external efforts in terms of population biobanks, like we have heard several times here, and I'm going to give a few examples of those. And actually, we also wanted to give back and put as much data out there and as much insights out there as possible towards the community. And in the end, this has also benefited not just the community, but also obviously our portfolio. How? Well, through obviously target identification, biomarker discovery, but also like giving us deeper insights into the diseases that we're interested in. So how do we do this, or how do we actually do this discovery of insights at scale? Well, the biggest data cohort that we have been working with by far, as you might suspect, is the UK Biobank. And this actually has been really revolutionary for us in terms of just the breadth and the scale of the data that it has allowed us to actually leverage, coming all the way from the initial releases, way back, up to the latest release, which is the whole genome sequences of the full 500,000 individuals, obviously with linked healthcare clinical data and proteomic and imaging data for a subset of them. So obviously, this actually has made it very, very attractive for its use in drug discovery. You know, obviously the sample size, but also, for example, since this is a cohort recruited in, you know, in their 40s and 50s, this actually aligned very well with many of AstraZeneca's portfolio priorities, especially around cardiovascular, respiratory, some cancers, and even we have been getting insights into rare diseases, which is something that people might not have thought at the beginning, but we actually have shown that you can indeed get rare disease insights from the UK Biobank. And how do we think about this analytically in terms of the kind of analysis and the kind of insights that we want to get? Well, as everybody knows, the kind of first large-scale unbiased look at genetic data came from GWAS, in which people were looking at association of individual variants with particular phenotypes of interest. Historically, GWAS began with doing these very large-scale genotype studies with imputation to actually enrich the data set. And they give you insights pretty much along the genome, you know, coding, non-coding, and this is, as we know, a common variant-based approach, right? I mean, and this is actually meant to discover variants of small effects that are relatively common. So the next thing that people have been doing has you know what we call the XWAS, exome-wide association studies, in which essentially we're still doing variant-level associations, but we're focusing first of all on protein coding variants, which is what the exome gives us, and actually we given that we have sequencing data for all of these exons, well, we can actually then interrogate sort of like much rarer variants. And the thing that actually we have really focused on in AstraZeneca for the past few years has been in what we call collapsing analysis. So essentially, when you take all of these ultra-rare variants, that might only happen on, one or two subjects in these very large-scale cohorts, but when we intelligently aggregate them, we can actually substantially boost the power, as everybody who's done burden test knows, and actually, we can build all of these collapsing models to actually derive insights according to different kinds of variants, and actually this is a substantial amount of the insights that we have been deriving from the UK Biobank and from other large-scale cohorts. So what does this look like? Well, we published a few years ago this first large-scale PHEWAS, this phenotype association study, where for each of the genes in which we collapse the variants, we looked at essentially all of the possible phenotype associations coming from the UK Biobank. And this actually allowed us to retrieve quite a bit of both known and novel disease associations with particular genes, which we found very exciting. And we actually found, for example, significant associations that were enriched for drug targets. And this actually pointed us to many novel insights that we're still mining, and we're still trying to figure out the best way in utilizing these. Now, going one step further, we know that the exome is very enriched for disease-causing variants, but the exome represents, what, maybe 2% of the whole total genome, and then the natural question is, okay, what are we missing when we only focus on the exomes and then we don't take a look at the whole whole genome? And obviously the problem had been one of scale and technology, where whole genomes had not been widely as available as whole exomes. This is now being remedied, and a lot of the population biobanks are switching over to whole genome sequencing, which obviously is giving us much more power to detect insights. We heard from, from the work in Genomics England in their 100,000 Genomes project about the insights that they have been getting with non-coding variants. And so what does this look like? What do these novel insights look like when we look at not just the exomes, but actually the whole genome? So we actually ended up joining the 500,000 whole genome sequencing effort in the UK Biobank, along with other industry participants. And actually, we have been getting quite a bit of insights that we have been publishing related to this novel data that we only got essentially last year, and we're still trying to actually mine. And as you might imagine, when you take a look at kinds of variations that the whole genome gave us that the exome doesn't, then we get quite a few novel insights. For example, obviously that copy number variants contribute to human diversity and disease. We actually found quite a bit of novel disease associations when we do essentially exome or rather genome-wide collapsing analysis and phenotype collapsing analysis based on copy number variations. We were able to recapitulate some of the associations between copy number variants and diseases, but actually we were able to find quite a bit of novel insights that we're still mining and we're still in the process of transforming into novel efforts. So my colleague Zoe Zhou in the Center for Genomic Research presented this data at IHCC last year, and we're in the process of actually publishing that. So this is very exciting. Just a little plug on our upcoming work. If anybody is going to be at IHCC this year, we're going to be presenting some of the work that we've been doing on short tandem repeats in the UK Biobank. So this is also very exciting for us. So I want to touch then on how we're actually using this data to get insights into particular diseases of interest, and how are these insights translating into novel therapeutic strategies. And I'll give an example that we published for diabetes, where essentially the amount of data that we're now able to actually mine and harvest on our hands is enabling us to actually ask novel questions. I mean, for example, typically the questions that people have asked is, well, what are the variants and what are the genes that predispose you or that associate with disease? And obviously, this is very useful for diseases that have a genetic component. This is very useful for diagnostics, to get disease insights, to develop your preclinical models. Now, however, kind of a more interesting question from a drug development and drug and target discovery perspective is, well, what are the variants that actually protect you against disease? And this is actually important because because you can ask, well, what are the contributors actually that protect you, that actually give you, that predispose you to not having a disease? And actually, you can ask questions for that, for even diseases or etiologies that are not purely genetic. I mean, so for example, we can ask, well, why is it that there are individuals who have high risk of HIV that do not get infected? Why are there heavy smokers that do not get cancer? Why are there individuals with diabetes that do not progress to chronic kidney disease, for example? And this approach actually has led eventually to very promising drug targets. I mean, we just heard that the case of PCSK9 that was published way back in, what, 2006, right? I mean, so, but then how do we scale and how do we generalize this approach? And the exciting thing about asking these questions is that if you can get an answer to those, then the path to a therapeutic discovery and a drug target is actually a much more direct. Now, the problem when you ask these questions is that normally you're underpowered to actually get these answers. I mean, so that's why you need all of these very large biobanks. And even now with the UK Biobank, we don't have enough power to actually answer some of the questions that we would like to ask for some diseases, so which is why it's important, as we have been hearing in this conference, the importance of actually working across data sets, across biobanks on increasing diversity. I mean, this actually empowers us also to do these kinds of discoveries much easier. So, for the case of diabetes, we use this actually philosophy for discovering this MAP3K15 potential target. And essentially what we discovered when we did this collapsing variant analysis was that essentially loss of function variants of MAP3K15 gave you a protection against diabetes, which was quite significant, and it was allele dependent, and it's a very similar story from what we heard, just heard in the previous talk. And so this was very exciting. So this was, again, very, very exciting, and you see, for example, that the degree of protection actually varies with sort of like the allele dosage. So people actually who are homozygous for loss of function protein truncating variants in this gene actually are even more protected than people who have only one copy, who are more protected than people who have both copies. I mean, so this is also very, very exciting, and has essentially provided us with this kind of insights into a potential therapeutic approach that we would not have discovered if we hadn't had this kind of data at our disposal. Now, I want to conclude finally with how we're thinking about rare diseases, which is my area of focus, because again, you can use all of these large cohorts and these insights, not just for common disease insight discovery, but also for rare diseases, which are close to my heart. And again, this is where I've been focusing my career almost for the past decade. Now, the challenge in rare diseases is that there's a lot of them. Depending on how you count, there might be 7,000 or 10,000, depending on the rare disease classification that you use. And only very, very few of them have approved treatment. So the count is about only 5% so far. And even though in many cases, we do know the genetics. Coming up with approaches that are scalable and that are feasible is very, very hard. Sample sizes in rare diseases, as you might imagine, represent a challenge, and we are vastly underpowered to do some of the stuff that we do for common diseases if we just directly try to apply them in rare diseases. So we also have a problem as a field on how to actually develop treatments that address very small populations in a way that is scalable and where the economics make sense. So I don't think that as an industry we've cracked that code, if you will, but this is an area of interest, obviously, so that we can serve more patients. And in many cases, fundamentally, we don't understand yet the disease mechanisms. So one thing that we actually have been doing is try to figure out how to close the gap between the common and rare disease research. So rare diseases, as you might imagine, have earlier onset. They're primarily driven by rare genetic variants. Many of them, not all of them, are monogenic, and what we're discovering is that even for the monogenic diseases, the genetic picture is complex, and there's many times not a direct relation between the genotype and the phenotype, and this is very complicated, right? And this is also a focus of what I've been working on that I won't have time to present today, but actually this is an area where actually I've been very, very active. So one thing that actually we were able to do, which I want to conclude with, is an example where we actually had developed a drug that had nothing to do with a rare disease, and by understanding better the biological mechanisms, we were able to actually repurpose that drug into an approved treatment for a rare disease. So we had a small molecule, selumetinib, which is a MEK1 inhibitor, it was initially intended for cancer. It failed to show in the clinic significant improvements when we tried it in lung cancer and thyroid cancer. But by understanding the biology better, we found that there was this rare disease called NF1, neurofibromatosis 1, that actually shared quite a bit of common pathways and common molecular mechanisms with these cancers. So we were able to repurpose that drug. We were able to successfully run several clinical trials on that. And again, the principle was understanding the RAS pathway and the tumorigenesis process in NF1, and it showed significant benefits in terms of progression-free survival, and it's actually now an approved treatment for the drug. So this is very exciting, and we're trying to actually figure out how to actually scale these approaches and actually how to build more examples where we bridge the common disease and rare disease research. So just to summarize, large-scale genomics improves our drug pipeline success rates. I mean, we have shown it, and this is an area where we feel that we're only started. And as our data grows and as our insights grow, we will get more and more insights into drug discovery from these efforts in large-scale genomics. And finally, I just want to conclude saying that genomics offers us an endless stream of exciting opportunities to actually improve drug discovery, and we're very, very grateful that we're part of this journey. This is a very large team effort that I want to thank both at the Center of Genomic Research, at our therapeutic areas, and obviously through all of our partners and collaborators, some of which are here in the audience. So thank you very much, and I look forward to the discussion and to questions. Thank you.
Thank you, Guillermo. Okay. Ask questions, use Pigeonhole. Simple. Our last speaker of the day is Dr. Mark McCarthy from Genentech. Mark is the Executive Director of Human Genetics and a Principal Fellow at Genentech. He leads efforts to use human genetics to advance molecular understanding of health and disease, and to use those insights to develop novel therapeutic and preventative approaches across multiple disease areas. Mark?
Hi, everyone. I can't quite get used to the music. I think I'm at the Democratic National Convention. So it's great to be back in Singapore. And like the others who hadn't been here, I think this is my 9th or 10th visit. So it's great to be here. I had many collaborations in my former academic life in Oxford with folks here. So it's great to be back and to renew old friendships and see and make some new ones. So yes, what I thought I'd talk to you about was present a few vignettes that just illustrate how we're trying to use not just genetic, but generally larger omic data sets to support drug discovery in our organization. And the kind of approach is summarized on this slide. It's about generating data internally, but it's also about leveraging collaborations and external data. It's about gathering data of a bewildering and increasingly diverse set of domains. It's about using state-of-the-art computational approaches to make sense of all those data, and it's about then applying that to do all the things that we want to do in a pharma company around target discovery, but also about a number of ways of thinking about precision medicine, for example. And we know that these approaches can help us. We already heard from Guillermo the evidence that human genetics targets with human genetics evidence are more likely to be successfully prosecuted through drug discovery. There's some nice data now emerging that you can derive similar and orthogonal benefits from single-cell genomic data, for example. And we've long known about the value of proteomics, for example, as a tool for patient stratification. So with that in mind, I thought I'd just illustrate with 3 examples of the ways in which we've recently leveraged these kinds of data to support mostly target discovery stories that I'm going to tell you about, but the same similar kind of approaches can be leveraged to add insights in precision medicine. So at the basis of a human genetics department in a pharma organization, of course, we make use of a diverse set of human genetic data sets, and I tried to sort of graphically schematize what we collect and what we use here, and there's a diversity of data sets, some of which we generate internally, for example, by sequencing our clinical trials. So those tend to give us information on phenotypes to do with progression, response, and biomarkers, which are very important for us, and probably more important than the genetics of disease risk. We leverage a variety of collaborations to access larger data, often with longer longitudinal components. We're part of the Open Targets Consortium, which gives us access to a lot of functional and mechanistic data. And of course, we make use of data from an ever-growing number of biobanks that are either available publicly or which we're investing in. All of these have their benefits. Of course, as you go up the scale here, the data sets become larger. They tend to be multi-trait, but they're less granular at the phenotypic level. And a lot of what we want to do is to move up and down and triangulate across multiple data sets so that we make discoveries and can validate them and generalize them in this way. One big limitation, of course, as we've heard, is that much of this data emanates from individuals of European descent and only very slowly catching up in terms of the necessary diversity. So the first story is a neat one that just illustrates how you can bring together multiple data sets to make novel discoveries. Diabetic macular edema, as many of you will know, is a subphenotype of diabetic retinopathy. And although diabetic retinopathy has been quite heritable until recently, there was a nice GWAS meta-analysis published very, very recently, but very few replicated genes had emerged from GWAS. It was one of those areas where GWAS didn't seem to be working terrifically well. One reason for that might be because it's a more complex phenotype, and by stratifying the phenotype, we might get a more manageable and evident signal. So we were focused on diabetic macular edema here. The reason why we focused on this is that we had an agent, faricimab, which had been authorized for wet AMD, but was also evaluated in clinical trials for DME, turned out to be successful and has been approved for that as well. But as a result of that, because we sequenced individuals in our trials, we had a reasonably sized, about 1,500 individuals with DME, which is probably the largest data set of really well-characterized DME patients. And it was natural to do a GWAS, comparing that against a potpourri of other individuals with other diseases that have been included in our trials. And we were somewhat surprised to see, despite the relatively modest size, a couple of very strong signals, and the one on the right was particularly interesting because it was a coding variant, a gene called ApoL1, which many of you will be familiar with, and a couple of haplotypes or alleles seen in Africa have been very strongly associated there with a variety of nephropathic phenotype, most obviously FSGS, but also CKD. And these alleles have been selected for because they're protective against trypanosomiasis. So natural to have a look at what the ethnic composition, ancestral composition of our data sets was, and whether the signal that we detected for this ApoL1 variant was present in African descent individuals or not. We only had about 84, I think it was, individuals of African descent, but the signal was strong enough to see that we were actually detecting a relatively equivalent odds ratio across the individuals of both European, African, and other ancestral groups. So, first thing you do when you see a signal of that is you try to replicate it. We had some other trial data that came through. We were able to replicate it. We went to FinGen, where there's not a code for DME, but there is one for diabetic maculopathy. We found the same variant was associated about 10 to minus 5 in FinGen. So pretty conclusive evidence that a coding variant in ApoL1 was associated with DME across multiple ancestries. So where was the signal coming from? As I mentioned to you, it's pretty conventional in the diabetic retinopathy field to feel that you have to get your control should be people with diabetes who didn't get diabetic retinopathy. We obviously hadn't done that, but it was fairly easy to show that the signal that we're seeing in those trials was coming from the cases and not from the controls. Could it be that because our controls were not diabetic, we were just picking up a signal for diabetes? Well, that was very easy to discount because there were massive data sets out there, some of which we generated in my former life, to do GWAS lookups for both type 2 and type 1 diabetes. There's absolutely no signal for that variant there. Was it hitchhiking on diabetic nephropathy, given that ApoL1 phenotypes in African descent populations? And again, we were able to demonstrate that that wasn't the case. What seems to be going on here, if you look at the haplotypic level, you can see there are four major haplotypes that are represented transethnically. The bottom two are the two that are associated with the renal phenotypes in individuals of African descent, but those are completely absent in individuals of European descent. The variant that we'd picked up, which is the one in the box there, picks up that signal in African descent individuals, but as you can see, is also associated separately with an independent effect on both DME in Europeans. So that highlights that a coding variant in ApoL1 that's apparent across multiple ethnic groups is associated with diabetic macular edema and has incited interest in this as a therapeutic target. It's a nice illustration of how you need to work across multiple ancestral groups, and Lyndon made a great play of this, and we're delighted to be part of the T4C effort that Lyndon's spearheaded. In parallel, we've been thinking about other ways that we can extend beyond African American individuals and capture more of the genetic diversity that's present across the African continent. It's clear that just looking in people of African American descent is not going to give us both the genetic diversity and the environmental diversity that we'll see on the African continent. To that end, we've put together a consortium building on those other pre-competitive collaborations that pharma's been able to support in UK Biobank and FinGen to try and do something similar around African populations on the continent of Africa, and as well as building new data sets to support infrastructure development and other capacity building on the African continent. It's clear that if we're going to do this, if pharma is going to build more equitable and sustainable partnerships, it needs to be done in ways that are very different to the way that we may have interacted previously with investment and leadership from African investigators. We've partnered with a number of African investigators, most notably Nikki Mulder and Christian Happy, who have been spearheading an effort to build genomic centers of excellence in Africa that many of you will be familiar with, which I don't have time to go into before. And we've worked with them and with others on the continent to build up sample sets that we can start to think about whole genome sequencing in, to build up sequencing infrastructure, to build support for informatics, to build support for training, to work with transnational agencies on the African continent to build policy to support genomic health. The goal here being to build a reference data set of 50,000 sequences diversely across the African continent, much of the data generated on the African continent, analyzed and processed on the African continent, and available preferentially to African researchers. So the second example I'll give you, it comes from the intersection of genetics and proteomics. And many of you will be familiar with the pharma proteomics project. I forget what the PPP stands for, but this is the effort to do large-scale proteomics, 3,000 proteins on the Olink platform, which is performed in about 50,000 individuals from the UK Biobank. And there are plans now to extend that out to the whole UK Biobank population. We were interested in working out to what extent we could use proteomics as a tool for a target identification. And we focused initially on asthma. If you look in the 54,000 individuals on whom we had both proteomics and genetic data, there were about 4,000 cases of asthma and 22,000 controls that we could identify. So a rich data set that we could start to mine for associations with proteomic data. Now, if you do a straightforward analysis, just take those 4,000 cases of asthma and those 22,000 controls and do a differential abundance, plasma protein abundance, you get over 600 signals come up positive. So 20% of the proteome shows significant differences between asthma cases and controls. And that's probably a mixture of things going on. Some of those changes will be causal, which are the ones you really want to find if you're interested in target identification. Some of them will be consequential. Some of them just represent confounding. So how can you split those apart? You can adjust for known risk factors like BMI and smoking. That's a bit of a dangerous thing to do because you may actually be adjusting out some of the causal pathways you're trying to find. You can try and look at the differences between incidental and prevalent cases in UK Biobank. That wasn't particularly successful. Or you can use genetics and do an instrumental analysis, and that's what we chose to do. So essentially, in those individuals in whom we had both genetics and proteomic data, we built genetic models of so what we term genetically predicted protein levels. In other words, polygenic scores for each of those protein levels, including both cis and trans signals. And then we took those out into the remainder of UK Biobank, where again, we had nearly 50,000 cases and 250,000 controls. And so that even though those predictors were only capturing a small percentage of the variation in those proteins, for many of the proteins, about 1,400 of them, we had pretty good instruments. And when we did this and looked for which of those proteins does the genetics that predicts those protein levels itself be associated with asthma, we came up with 70 hits. And I've just shown here the 16 that came up from purely cis analyses. And this is a mixture of genes and proteins that had previously been implicated in asthma, some of which were already therapeutic targets, and a number of other new proteins that previously hadn't been implicated in asthma pathogenesis and become starting points for thinking about novel therapeutic discovery. Just to give you one example, a TLR1 variation, so variants that are predictive of TLR1 levels in the plasma are predictive of asthma. And this signal co-localizes with a signal for IL-27 that suggests, particularly when you do mediation analysis, that TLR1 levels are mediating their effect on asthma risk through an intermediate effect on IL-27. And then just finally, let me tell you about some of the ways that we're thinking about going forward. I mean, we are at a really interesting time now where the volume and data that we're able to collect from human subjects, as you all know, is vastly increasing. That means we can shift from reliance historically on preclinical models and so on to doing much more of our discovery based on human biology. We have the fortunate position of assays that we can perform at large scale whilst maintaining high resolution. We no longer have to trade off quality and quantity. We can get both of them at the same time. And we have computational tools, particularly in the AI/ML space, that allow us to make sense of those vast amounts of data that emanate. And the amounts of data are vast. If you think about the number of cell types in the body, if you think about the number of genes, the number of variant combinations, the number of possible antibody sequences you could generate as therapeutic antibodies, the number of potentially druggable small molecules, these numbers are astronomical. They're often bigger than the number of atoms in the universe, so there's no way that we can experimentally enumerate all the possibilities. There's no way we can do some of this even computationally. So we have to think about ways of representing those data, building models so that we can work through the possibilities in a manageable way. So we're leveraging a lot of AML tools under the sort of general sobriquet of lab in a loop, where we generate data, build a model, test the model, and then generate more data to iteratively improve the model, and using this as a tool for both target discovery, for thinking about novel therapeutics, and so on. I'll just give you one example, and this relates to increasingly view that a very useful way to think about a lot of biology and a lot of the therapeutic avenues that we can take is at the level of the cell. And identifying pathogenic cell types, understanding toxicities, understanding bispecific targets, and so on. All of that relies on understanding the cellular mechanisms involved in disease and how you might manipulate them for therapeutic benefit. So if you're going to make sense of the world of, for example, single-cell data, how are you going to make sense of it? So one of the things that we've been doing is scraping all of the single-cell data that we can find in various repositories, as well as data we've generated ourselves. This slide's a little bit out of date, but now, so far, we've collected over 300 million single-cell RNA-seq data points, in other words, data from over 300 million cells, I should say, from several thousand studies, collated all that data, brought it together, and harmonized it, and then started to employ AI/ML tools to organize those data, to categorize those data, to label the data, and very importantly, to give us a mechanism for being able to query the data. So one way of thinking about this is a bit like a Google image reverse search, where you have a picture of somebody, you don't know who it is, you get Google to tell you what it is by comparison with all the other images in the database. And we can do something like that very efficiently now with a cell that you might identify from an experiment, and trying to work out, well, what the hell does that cell look like? And by reference to those other data sets, what is it? Because many of those other data sets will have labels that allow you to transfer the label to those cells and annotate that cell. So just to give you one example of one of the ways in which we've used this, we were recently interested in new therapies for idiopathic pulmonary fibrosis, particularly vixarelimab, which we brought in as an agent against oncostatin M. And the reason that we were interested in this is because there's a strong biological belief that part of the mediating mechanisms for the generation of IPF, which is a truly horrible disease, is through damage in the lung that leads to inflammatory cell recruitment and fibroblasts that express OSMR that lead to fibrosis. So that was fine. We had an agent that we started to put into phase 2 trials to see whether it performed in IPF, but natural to ask whether these cells were present in any other disease states. So the plot on the right shows you what you get when you take all the cells that we have in this compendium, which are organized according to the cell, the tissue from which they emanate, and look for enrichment of these OSMR expressing fibroblasts, these inflammatory fibroblasts, across those other data sets. And the darkness of the color is telling you the frequency, the prevalence of those cells. So you can see, we see a hit over in the sort of bottom right, about 4 o'clock there, in many of the samples that we have categorized as coming from lung, and particularly for those that have various forms of idiopathic lung disease like IPF. But we also saw a hit in some of the samples that we had from gut. So that raised the possibility that as well as in IPF, OSM expressing fibroblasts might be mediating some of the pathology in inflammatory bowel disease. And that was corroborated because there are a number of SNPs near the OSMR receptor that are associated with various forms of IBD. And that gave us enough confidence, without doing any preclinical work or any further biology, to extend the trial portfolio to IBD, and we'll obviously see how that works. So a big part of what we're thinking about now is this kind of integration of large-scale data generation, algorithm development using AML tools, and then further experimentation to build the ever-increasing performance of these models with the ultimate objective, to some extent, that they become self-driving. We're capitalizing on the opportunities that come from no longer having to trade quality and quantity in the ways that we had to do before. That's a lesson we learned a long time ago in human genetics when the GWAS era came on, but it's now starting to play out in many other omics fields. And a lot of this is also powered by innovation in the AML space. So I've tried to give you 3 examples of ways in which we're working to bring multiple data sets together, to mine data sets, to combine data sets, to combine data of various different domains, to employ large-scale and state-of-the-art computational tools to try and make sense of these data, and how these can play out in terms of new approaches to identify novel targets. But I could have equally shown you some examples that points towards ways of thinking about precision medicine. So with that, let me just thank some of the folks who've contributed to the work, some of whom are in the audience. It's great to see you, and thank you for your time.
Okay. Let's get started. First question. Selecting it here. Thought I knew how this worked. Aha. I got it. So, how much have new AI methods entered the drug discovery process? For example, foundational models on single-cell gene expression data, transformer LLM on GWAS, genomes, and generative enhancers. Who'd like to start?
I can maybe start, because I made a start on that towards the end of my talk. I mean, maybe we've drunk the Kool-Aid quite a lot in my company, but we're investing hugely in this space because, for the reasons that I stated, if we're really going to leverage these data sets that we can now generate and make sense of very complex biology, I think we need these tools. We're doing all of these and a bunch more. I mean, I think one thing that's important to say in qualifying this is it's not just about having the AML capabilities. Unless you feed them with large amounts of data of high quality, then you're not really going to get very far. Sometimes those data are available out there, as we demonstrated with the single-cell RNA-seq, and we've found the foundational model that we've generated from that endlessly useful in lots of different ways other than the ones that I just discussed. But I think in other areas we are just simply all going to have to roll up our sleeves and generate data at the kind of scale that allows these methods to really do their thing.
Yeah, absolutely, and this is work in progress. And just to mention 2 very brief examples that we published, we published this tool called MantisML that essentially tried to find novel gene-to-disease associations based on machine learning. And the problem with gene-to-disease associations is that you have a lot of positive examples, but you have like very few negative examples. So we had to really get creative on how we architected the whole machine learning model to actually build these negative examples so that the model could be trained, right? And so you have to be able not just to actually get the right data, but actually build the right architecture into the model, considering what data you have available. And this is something important, right? I mean, that this is not just like a blind vacuum cleaner of data that will automatically ingest it and give you insights. I mean, you have to really think about the problem and what data you have and how you actually want to do it. The other example that we just published a preprint and we're in the process of doing the peer review process is this enhanced biomarker predictor based on proteomic data. And so we're trying to integrate the genetic and the proteomic data to actually predict new biomarkers for diseases. And obviously this is very relevant with, for example, the plasma proteomics project in the UK Biobank, et cetera. And again, the problem is that the data is very noisy. You have to actually figure out you know, how to normalize it and how to actually build solid models that in many cases will be very, very prone to not be able to generalize very well. So you have to like figure out, okay, you know, how am I going to normalize my model? How do I actually restrict the parameter space so that I don't get like, you know, this overtraining, right? I mean, so again, you have to be really, really thinking about the model and the data that you have available and the problem that you're trying to solve so that you actually get useful results.
Yeah, I won't add, but just to reiterate that, you know, it's the data that you input that's important for this. And as we generate more data, better models, we have better AI tools.
Lyndon, this question's for you. Will the Together for Change data be put in the public domain?
No. The whole purpose of this is to provide the data to these schools and let them have the advocacy and the power to share the data with whom they would like to share it with. If we put it into the public domain, organizations, I don't want to call your names, will analyze the data very quickly, publish the papers, and get all the grant funding. The whole point of this is to create a unique database for these schools to become players in the genetics field. So we're not restricting anybody from getting the data. We're encouraging you to collaborate with these schools.
Thank you. Thank you. Next question. Nope, no, I already did. Sorry. What is the social responsibility of pharma companies to make new drugs accessible to people from low and middle income countries, especially as they start using data from underrepresented populations for new target discovery? Who'd like to start?
You're looking at me? Yeah. No, this is a great question, and there are lots of ways of approaching it. Clearly, part of the reason that pharma wants to have access to data from individuals that are more fully representative of genetic diversity is to enhance target discovery, but an equally important part, and actually a stronger driver for our company, is to make sure that the medicines that we're already developing, more and more of which, as they come to market, we're going to be expected to understand how their efficacy and their safety maps onto individual variation in genetics and other demographic features. And we do not want to be in the place in a decade from now where we have that information for you if you live in Hamburg or you live in Houston, but we don't have it if you live in Harare or anywhere else beginning with H, Hanoi, let's say. So I think it's really important that we think about how we can access data sets responsibly from as wide a variety of individuals as possible, not purely to make new drugs that we can sell in the wealthy parts of the world, but so that we can develop drugs that meet the needs of those populations and also ensure that the medicines we're developing anyway are going to be as safe and effective in those populations and everybody across the world. That's an important part of the motivation. And that's part of the social responsibility, is to make sure that those medicines we know are as safe as possible. I mean, clearly there are a number of issues in terms of the relative economics of countries and so on that we have to engage with. It's a very unequal world. And I know we and other pharma companies are really committed to finding ways of thinking about benefit sharing, of which this is one example. But there are many other ways in which we can share the benefits and make sure that what we do is responsible and equitable and sustainable.
Absolutely, and there's relatively little that I can add to that, Mark. But the one thing that I would indeed want to add is that the more we understand the genetics of underserved population, the safer and the more effective treatments we will be able to develop. And obviously this is going to benefit everybody in the world. One thing that I didn't have time to touch upon today is that we're using also these large-scale genetic databases to find potential safety signals. I mean, essentially, you know, when genes are associated with potential safety indications. And the problem with not having diverse data sets is that you don't see these potential safety signals in other populations. And this is something that we have to be really, really cognizant of. So the drive for more diversity is will actually give us also safer treatments and more effective ones.
Yeah, I would add a different angle and say that, you know, there is a social responsibility for our pharma companies to ensure that drugs are in LMICs. And we got to help them to build the infrastructure for a lot of these types of drugs because, you know, there is an infrastructure issue here as well as the diversity and the effectiveness of these drugs. But think about 10 years from now, we're going to have a lot of genetic medicines, a lot of medicines that, you know, more biologics. You know, these are not just pills that you can, you know, send over in a regular, you know, airplane. You have to really think about the infrastructure of getting these types of drugs to patients in these different countries. So this is why I like the Get Together for Change initiative, and this is why this initiative is 10 years, because we're going to have the opportunity, and I would say for the LMIC countries represented here, to, when you are building those collaborations with companies like ours, you know, talk about these kinds of things and say, okay, look, we'll do these types of collaborations with you, but let's have conversations as well about how to get drugs to our country, how to change policies in our country, because there's a lot of political things that need to happen in a lot of these countries as well. So we got to start having these conversations and talking this through, talk through the problems that we have, because there's a lot of drugs we have right now that can be in a lot of these countries, but, you know, there's an infrastructure issue, there's politics involved, and I think if folks get in a room and start talking about these things, we can solve these problems.
Thanks, Lyndon. Next question. We've achieved a lot for human genomics. What is your review and vision on the human microbiome, metagenomics, and drug research? Who'd like to start with this one?
That is a great question, and obviously I'm not a microbiome expert. I know very, very little about that. I think that is a vastly unexplored territory, and we need to be better at actually mining it. I know obviously that there are companies that do that, and that they're really pioneers in the space. To be honest, we're not one of those, but this is certainly an area that we shouldn't neglect in the future, I would just say.
Yeah, it's an area of definite research. You know, we need a lot more research in this space. And, you know, we're trying to even figure out how the human genome works. And now we're layering on other genomes. So I do think it's important, but, you know, I think as we sequence more microbiomes and so forth, we're gonna figure this out just like we did with the human genome.
Yeah, the only thing I would add there, I mean, it's not my area of expertise at all, but I'm certainly aware that the data that I've seen is that the metagenomic profile that you get in the samples that normally used for this are not very representative of what's happening in the crypts in the gut, for example. So we need just much better ways of sampling the data and actually finding the bits of the microbiome that probably matter more than what you get from your average stool sample.
Okay, beyond genomics, not the company, the question. What is the pharma view of other omics in drug development? Which omics is of the most interest and why?
All of the omics are important.
Yeah.
It's just, just like the sequencing technologies have allowed us to, sequence millions of people now, right? The other omics that we're talking about here, like proteomics that Mark talked about, is starting to get there. And, I think as we, refine a lot of those technologies like proteomics and metabolomics and so forth, I think all of those are going to be hugely beneficial to making new discoveries and understanding biology.
Yeah, absolutely. And I don't think I could name one that we're not interested in. I mean, I think that all of them are very, very relevant. I mean, we use bulk RNA all the time. We're doing more and more single-cell RNA, and the more you do single-cell RNA, you realize, why was I doing bulk RNA? I mean, what was I doing? I mean, how could I get insights now that we have all of this wonderful data available, right? Obviously, we have done a lot of proteomics. We haven't done that much in terms, for example, of epigenetics and studying, for example, patterns of methylation and other epigenetic changes. I think that this is vastly unexplored, and this is very, very important. And we're in the process of actually figuring out how to derive insights and how to actually get better at interpreting that signal.
Yeah, I agree. All the above. And one thing that hasn't been mentioned, the more bulk, the more single-cell RNA-seq you do, the more you wonder about why you did bulk, but the more you want to do spatial, because I think the insights that come from actually understanding how those cells co-localize and how they sit, and the cells with different single-cell profiles relate, for example, to proximity to neurofibrinolytic tangles in the case of Alzheimer's and other pathology is probably what's going to deliver some fundamental insights into disease biology.
Next question. Could you think of ways pharmas could share the profit made based on biobank data? Even 1% could help a lot in this area.
I agree. But here's the catch. And first of all, I do agree. I think we have to figure out the benefit sharing models of profit sharing and so forth. But we gotta be fair across all of the studies that have been done and that will be done. But two, I think I can see the headlines, right? Regeneron gets 99% and University X gets 1%. So we gotta figure that out as well. That make it where there is some benefit sharing and it's fair.
I think you also have to consider the time between a target discovery and a drug on the market too.
And the low probability of success, right? I mean, I think it's very easy to say, because you've analyzed this data set, PCSK9 story is the one that resonates, right? But if there were lots of PCSK9 stories, I can tell you, you'd hear about lots of other examples other than PCSK9. There's a reason why. PCSK9 is trotted out every time. As I showed with the ApoL1 example there, right? It took multiple different data sets to come together to build a picture. And it's rarely the case that any single one, so it's rare that you can say, oh, we only made this discovery because of this finding. And then you've got the situation that anything that you find as a genetic target's maybe got 5% chance of making it to the clinic in 15 or 20 years' time, if you're lucky. So I think, if we're really going to solve some of the problems that exist now, we've got to find other ways of benefit sharing and so on, not simply profit sharing based off or reduction in price on the drugs that may or may not emerge from a given finding. There's other ways to think about benefit sharing.
Yeah, absolutely. And the problem that we have as an industry is that we're not paying for all of the successes, we're also paying for the failures, right? So it's you know, for every discovery that is successful, I mean, you have like a number of them that will not make it for causes that are not related to necessarily just the basic biological insight, right? I mean, it could be something in your molecule, it could be many other things, right?
But just to reiterate, because I think, you know, may get the impression that we're trying to be evasive here. I think both Lyndon in Together for Change, which we're all members of, and this is the work that we're trying to do in Africa, we absolutely recognize that these things have to be done differently to a historical. You know, none of us would sign up for helicopter genomics. None of us wants to do things in that kind of old way. There are obviously tensions when North meets South. There are tensions when profit meets not for profit. And we're all trying to find ways of doing this in a better way, a more equitable, fairer, and sustainable way. You know, not everybody agree that we're doing everything that we can, but we're certainly, I think all of us, trying to move the needle here and get to a happier place where everybody can benefit from the research.
Absolutely.
Yeah, and Together for Change, I'm glad you brought that up, Mark, is, you know, we're kind of paying a little forward. We're trying to help, and we have a grant component, a part of this initiative. So part of the funding went towards sequencing 500,000 participants, but the other part of the funding went towards grants for building the DNA Learning Center and building K-12 types of programs, the genetic counseling training program. So it's a model. It's not solving this issue of benefit sharing. Trust me, I understand that. But us getting together for 10 years and having that conversation, and if our companies are successful enough to identify new targets and take a molecule into the clinic based on those genetic findings, we're going to want to have conversations about how we can help this community because it's successful. So that's why these types of partnerships like T4C is designed for those conversations to help with that question of what can we do to build more, you know, benefit sharing models. We don't know the answer. We don't have the answer, but we can figure it out together because we need to understand what the community needs are and how we can do that and be successful together.
That's the end of this session. I'd like to just thank all the speakers for the questions and the answers you've given us. You seem all very sincere and it's hopeful.
Thank you, Scott. Thank you.